Welcome guys to our little online lab overview. Today we're going to be talking about antimicrobial resistance and the zone inhibition test.
Yeah.
So we're going to start by breaking down some of these terms and getting into some of their properties. Antimicrobial resistance. Antimicrobials are substances that are used to control the rapid growth of bacteria by inhibiting their growth and slowing down their rate of division. Antimicrobials can take the form of antibiotics, disinfectants, and antiseptics. Antimicrobial resistance takes place when bacteria or other pathogens change over time and no longer to respond to the medicines. So resistance can occur in bacteria partially or have a full resistance to an antibiotic. And we'll see that in this lab. The zone of inhibition test is used as a measure of susceptibility to excess antimicrobial activity. It is also known as the Kirby-Bauer test, antimicrobial susceptibility test, diffusion test, or Auger diffusion test. So as you can see in this illustration, it depicts what you'd come across in this lab procedural test. On the left side, we have a petri dish that is freshly inoculated, so there's no growth yet, and on it are some antibiotic discs. Now, the purpose of the experiment is to identify the effectiveness of different microbial agents. So what you'll see in the petri dish on the right is bacterial growth represented by the lines there. That white around it is the zone of inhibition, and that's no growth at all. And that's the kind of thing that we're looking for in this experiment. So in this lab, we're going to be observing the effects of several different antimicrobials on 3 different bacteria as listed here. The 3 bacteria that we're going to be using is Haemophilus influenzae, Staphylococcus aureus, and Streptococcus pneumoniae. And our antimicrobials here on the left side include these 7. 1, we have our sterile filter paper for control. 2, antibacterial soap. 3, household bleach. 4, household disinfectant. 5, penicillin. 6, amoxicillin. 7, erythromycin. And these are numbered just to help you see like how everything was laid out on the petri dish. They will be in the exact same order, and I'll show you that in a minute. Prior to the start of the experiment 6, which again investigates the effect of antimicrobial agents on various bacteria, it was hypothesized that out of the 6 antimicrobial agents and 1 control, that bleach would be the most effective and have the largest zone of inhibition against the bacteria. And this thinking was kind of due to the knowledge that of the characteristics of household bleach and its use in disinfecting. Another hypothesis posed in this experiment was whether or not the bacteria would be resistive, and therefore if the zone of inhibition was independent or dependent on antibiotics. And with that, if bacteria were able to grow within the zone of inhibition, what traits did the bacteria possess? Another thing to consider would be how will the bacteria be affected by various antimicrobial agents, and what does the size of their zone of inhibition tell us? So now that we kind of know what we're getting into, let's jump into the materials. So here we have the 3 types of bacteria we're going to be using in this experiment. And like I said, they were listed below: Haemophilus influenzae, Staphylococcus aureus, and Streptococcus pneumoniae. And we also have our 6 antimicrobials. And don't forget about our sterile control. It's not listed here, but it's basically just the disc soaked in water, so I just left that out. And we also have a few other supplies. So getting into when I actually conducted the experiment, starting off I began by inoculating our agar plate using a sterile loop. with each of the bacteria. So they're going to look something like this, the image here on the right. The quadrant method is something that I've always used. It's what I was taught, so I kind of stuck with it. And of course, these samples were properly labeled for easy identification. And again, we're using these bacteria. And each bacteria would have its own plate, of course. Next, students conducting the experiment placed a single paper disc from each tube of antimicrobials onto all 3 of the inoculated discs, which is your image on the right. Like I said, they're in a specific order. As you can see, they're numbered. 1, our sterile filter paper as our control. 2 would be our antimicrobial soap. 3 would be our household bleach. 4, household disinfectant. 5, penicillin. 6, amoxicillin. And 7, erythromycin. So these samples were placed into the incubator for 24 hours after inoculated. So I just wanted to bring this up, but these are some of the things that you should kind of keep in mind throughout the experiment. You always want to make sure that you're carrying out these tests with precautions and just pay attention, right? Like you should take steps to prevent contamination, so not making contact with not sterile materials with sterile. You want to close the tubes and our culture plates when not immediately using, and also just mark everything so items don't get mixed up, because of course this will affect your results. Another thing, you want to pay close attention to the time. You don't want to leave your cultures in the incubator for too long, and just make sure to keep accuracy in your measurements. Like use the same ruler, because sometimes they vary by just a little bit, but that can throw you off your measurements. And the same with other materials. If you're going to use them with each one of your cultures, make sure you sanitize them in between. And yeah, keep the same materials because you want to ensure similarity in all your outcomes. Overall, you just should keep a close record of things in the lab. Labeling is also very, very important. So make sure that you can label them and distinguish them to prevent mixing them up. Fast forwarding to our 24 hours, our cultures show obvious signs of growth, and we can see the zones of inhibition surrounding our antimicrobials, and these cultures have been observed and are measured in millimeters. So I just want you guys to notice this tan portion here in each one of these petri dishes. That is the zone of inhibition. And the one in the middle, that is our control, our sterile paper dish, so of course it has no zone of inhibition. So our measurements are recorded here. So based on the results, we can conclude that the different antimicrobials showed varying degrees of inhibition, and this is due to the contrasting characteristics of bacteria and its resistance to certain substances. As you can see from our cultures and the chart, neither one of the bacteria were fully resistant to any of the antimicrobials. All diameters had at least a minimum of 10 millimeters, with only 4 of them reaching into the maxes of 30 or more. So this is our slide for Haemophilus influenzae. Some things about it: it can cause ear infections and is known as the leading cause of bacterial meningitis in children under 4 years of age. The results for this bacteria are highlighted in the chart below, and on the right you can see the zones of inhibition for this specific bacteria. Staphylococcus aureus can cause superficial and deep abscesses, wound infection, infection of various internal organs in humans, cause food poisoning and toxic shock syndromes, and is the principal cause of cow mastitis that affects domestic cows. And just like the slide before, the highlighted portion is the diameter in millimeters for this specific bacteria, along with the corresponding image on the right. And the stain for Streptococcus pneumoniae. It can cause pneumococcal pneumonia, has spherical cells that are arranged in chains, and is the organism in which function of DNA was discovered. So that's just something about that, and their results are recorded in the chart as well, the highlighted portion, image on the right. Conclusion. This experiment's purpose was to compare the effectiveness of different antimicrobial agents on bacteria. The bacteria that we used, again, were Staphylococcus aureus, Haemophilus influenzae, and Streptococcus pneumoniae. Some of our antimicrobials include 1. The sterile paper disc, 2. Antibacterial soap, 3. Household bleach, 4. Household disinfectant, 5. Penicillin, 6. Amoxicillin, and 7. Erythromycin. From this experiment, we can conclude the affirmation of one of our hypotheses. It was hypothesized that out of the 6 antimicrobial agents and 1 control factor, that bleach would be the most effective and have the largest zone of inhibition. So we can make this conclusion based off the averages taken from diameters of each of the antimicrobial agents against bacteria, which is highlighted in this particular graph. It shows the household bleach. All of the diameters associated with bleach averaged around 30 millimeters, and the other ones averaged out to a range from 15 to 24. Another hypothesis made in this experiment was whether or not the zone of inhibition was dependent or independent on antimicrobial resistance. Through lab procedural test results recorded in the table, we can conclude that the zone of inhibition was fully dependent. All the samples produced a zone of inhibition with diameters greater than 0 millimeters. And finally, how will bacteria be affected by various antimicrobial agents, and what does the size of their zone of inhibition tell us? To answer this question, we can just simply look at the charts and its values and how they differ. The diameter of the zone of inhibition is related to the present levels of antimicrobial activity, where the larger the zone, the more antimicrobials are present. So keep that in mind. Alright, and that is our last slide. Thank you guys so much for watching, and have a good night.